Alright, you guys, let us go ahead and talk about the microscope. So hopefully you watched the previous video that was talking about cells, the difference between prokaryotic and eukaryotic cells. So now we're going to take a look at and talk about the microscope, how it works. You are going to be required to know the parts of it and what those parts do. I know it's not as fun because we don't get to actually use a microscope, but we will try our best to still have some fun, even if it's just virtual, right? So the main idea here is that we need to be able to make proper scientific observations, even if you are not the one like actually handling the microscope right now, right? So I'm not really gonna talk about it too much because we talked about it in a previous PowerPoint about cells, but as we talked about yesterday, we know hopefully that when you're doing the scientific method, you have to first start with observations, right? So for our class in particular, we are practicing how to be and think like a scientist without all the equipment. So observations are going to be one of the most important skills that you will develop as a scientist. And again, I say as a scientist, but really in any other career that you go into, observations are going to be really important, right? Even just observing your surroundings, that's very, very important. So back in the day, right? Back in the day, before we had any cameras, any of that fancy video, stuff like that, in order to collect data, they would have to draw it, right? So all of the pictures you see here are hand-drawn pictures. So not every scientist was a great artist. So if you were maybe not so great at drawing and looking at the stuff, then maybe you would hire an artist to come with you and, draw what it is that you want to draw, right? Because like I said, not every scientist is an artist. Sometimes they are, but not always. But the drawings are very, very, very accurate. And if you guys can see, like even this beetle picture right here, it has different like angles, right? Different ways of looking at it. Even something like this bird picture right here, right? They have a front view, they have a side view, they even have like this color palette right here. And even when they can't draw it properly, they'll put these little notes on it, kind of like animators, right? So if you guys, any of you guys are an art major or hoping to work for animation studios in the future, it's the same thing. You're trying to create this image that can speak for itself, essentially, right? Speak for itself. So we talked about it a little bit ago, but microscopes were very, very, very important in giving us insight of the microscopic world. So here's the one by Anton van Leeuwenhoek, and you can see this lady right here is using it, and it just kind of looks like a tiny magnifying glass, but that was the start of the start of something new. If you want to refer to High School Musical here, they couldn't see much. They could see like simple figures like this bigger, right? But again, it's just having this idea of there's something more out there than the big organisms was really a revolutionary thought, right? So we talked about this in the previous video. We have this old school microscope that Robert Hooke made. This is the one we will be talking about, but there's a lot of fancy microscopes out there. Like I said, this is one of the things that really made me fall in love with the cell biology major, is that there's just this whole other world where you see all of these tiny things that your eye can't see. So to me, that's really, really cool. This one is one of my favorite types of microscopy. It's called fluorescent microscopy. I probably spelled that wrong. Fluorescent microscopy. You put fluorescent dyes to pinpoint different parts. So if you take a look at this legend right here, it says blue is DNA, red is mitochondria, green is actin microfilaments. So these special dyes will go to those structures, light them up depending on what color they are, and then we can visualize it, right? when we look at cell samples like in real life, they're not colored. Most of them are clear, to be honest, right? So to be able to see what they look like, you have to put some kind of colored dye on it. Some of them, again, more powerful than others in order for us to see things, right? But if I just like straight up took a cell out of my body, it's not gonna be any color. It'll be clear. Except if you're looking for plant cells. Plant cells, because they are green, some of their cells are green, right? I would say most of their cells are green. But here are a couple more cool microscopes that I would like to point out to you. The first one is called TEM, or transmission electron microscopy. This is being able to see images inside the cell. So that is your keyword, right? Inside the cell. Because I cannot take, like, for example, I can't take a liver sample, a liver cell, and use my teeny tiny little microscopic knives to cut it open, right? There is no such thing as a teeny tiny little microscopic knife. So what you would have to do is just cut off the sample, put it into this transmission electron microscope, and then you can see the inside. So that's this one right here. So inside meaning if I cut, what example? So like think about an orange, right? If I cut an orange in half and I'm looking right at it, the orange would look like this. Sorry, that's an ugly orange, but you guys know what I mean, right? So it's kind of like that. I cut this cell in half and I'm looking right at it. That's what the transmission electron microscope is showing you, right? The other one that's super cool is the SEM, scanning electron microscopy. This is the image of surfaces. So that's this like buggy eye creature right here, right? So just like the word says, so how I remember this, it says scanning, kind of like you're scanning something on the computer, right? It does the little thing where it scans over your document and then you have an image of the document, right? So same thing here, like this image right here, I don't want to get too technical on you guys, but they're shooting electrons at it, and then the electrons will give an image back to you. So then it looks something like this. So you can see all the details of the surface, but I cannot see inside the bug. If I want to see inside the bug, then I would have to put it into this transmission electron microscope. If I want just the surface of it, then it's the scanning one, right? So yes, you are going to need to differentiate between the two. If you're interested in stuff like that, let me know. I can give you more resources, but for our sake, we'll just leave it as is. This is what we need to deal with, the light microscope, right? It's called a compound or a light microscope. You do need to know the parts, right? So I'm gonna delete some of the ones here that you don't need to know, but there's like 2 of them. So this one you don't need to know. You don't need to know this one. Let's see. And I think that's it. Oh, this one. You don't need to know that one. And I think that's it. So like I said, not much, right? I'm going to try, I think some of your videos that you have on Canvas will show you how it works if it's right in front of you, right? And unfortunately, that's just kind of the downside of doing an online class is we don't get to play with these things. But I would still take a look at the videos that are linked on your Canvas so you can at least have a feel of what it looks like, right? It's not going to be as fun or high tech, but at least you get to see it. So I'm going to put a star right here. So you need to know how to label the parts. Label the parts and what they do, right? And you won't be able to really figure out what they do without going through some of those animations that are on your Canvas. So please go take a look at those virtual assignments. The main idea here on this light microscope is it actually uses light. That is the main point, right? But the problem is it's a bunch of mirrors and lenses, right? So if you can see here from the picture, the light needs to hit each of the lenses properly in order for an image to show up. So this, pick a different color. This is where the light comes from the bottom. It's like a light bulb, right? So the light needs to hit, that's not a good color. The light needs to hit every single one of these lenses, right? And travel outward for your eyes to see it. If there's something that's not lined up, you won't be able to see anything. Usually it's just black, right? And then I would come over to you and you'd be like, I can't see anything. And all I have to do is like click over one thing and then it's all fixed, right? So it's very, very important that all of these are lined up. Otherwise, you won't see any of the images, right? So it's this one right here that I'm talking about. Okay. This is another one of your learning goals. So I'm going to highlight or put a star on this slide because you need to know the difference between resolution and magnification, right? This might be self-explanatory, but we're going to talk about it in the context of a microscope, so it makes a little bit more sense, right? So resolution right here, it's the ability to distinguish 2 points as being distinct. So it's kind of like when you take a look at like a picture, right? So if I don't know, if I'm going to a Taylor Swift concert, right, and I told you I'm going to a Taylor Swift concert, but all I have is a blurry picture of Taylor Swift, you're not going to know that I actually went there, right? The resolution of your camera, the clearer it is, the higher the resolution. So for some of you guys who have the fancy phones, right, like the upgraded top of the line phones, you will have high resolution like this one, so you can see every pixel really clearly, right? Whereas something like poor resolution, that'll be, I don't know, like the first ever iPhone, right? The first ever iPhone compared to the iPhone now, definitely big difference in resolution, right? Or I don't know if you guys know what this is. I'm just old, but back in the day, we had flip phones that did not take very good pictures, right? So that would be poor resolution. And then we have something called magnification, and just like normal magnification, it's making the image bigger, right? So we're zooming it in. And again, a lot of the bacteria, a lot of the prokaryotes and cells that we are going to be studying are not seen with our naked eye. So we need to zoom in on it in order to see it, right? Otherwise, like, you know, if I was Robert Hooke or Anton van Leeuwenhoek and I started talking about cells and I don't have any image or any visual of what it looks like, people are going to think I'm crazy, right? Which I'm sure they probably felt that way at the beginning. The problem with magnification and resolution is they work against each other. So I'm going to write that down. Resolution and magnification, that's ugly, work against each other. They do not work together. They work against each other, right? That means that the more I zoom something in, the less clear it becomes. And you can do that on your phone, right? Like if I'm trying to take a picture of something far away, I have to zoom in on that image, which usually means that it becomes blurry, right? It might still be, you know, distinguishable, but blurrier than if I took it right in front of me. So this is an example right here of a microscope image, right? So you can see that you're magnifying the object, but the resolution is really bad, right? So this is a highly magnified image, right? But the resolution is really bad. So this last one right here is the best one because it's very sharp, very clear, right? And it's not even too pixely, it's nice and crisp. So that is the goal. This is the goal, right? So when you're using a microscope, that's something that you need to work on, and it's really a skill. It's trial and error. And I feel really, really bad that you guys don't get to play with the microscope because it's one of my favorite things. But it is something that you need to practice. It's not like, you know, I put a microscope in front of you and then you're pros right away, right? Here's another example. I'm pretty sure this is Professor Johnson's dog, right? So this first column of pictures right here is high res, high magnification, right? So high magnification because I'm able to zoom in on the dog's nose, right? And this one, I know that this one is high resolution because I can see the little bumpies on his nose, right? Whereas over here, this one is high magnification but low resolution. So it's the same one, I can zoom in on it, but this one, like if I wasn't looking at it, I might think that's an eyeball of an alien. and kind of like this, that's his eyeball. He's an alien, I don't know, right? But because it's so blurry, you can't see it, and that's the problem. Speaking of aliens, that's the problem with aliens. Most of the pictures that we get of aliens are very blurry, so it's very hard to distinguish between that. So it is the same when it comes to microscopes. If it's blurry, we won't be able to see anything, right? So we're going to talk a little bit about how the microscope uses its sizing, because that's the thing that we have to learn and we have to focus on in this virtual environment. So you're just going to pretend as if, you know, another scientist took the pictures of the microscope, but you are the one that needs to analyze the image, right? So that is just the story. That is your job in our little story that we're working on. So on the microscope, we have something called objective lenses. So let me just backtrack really quick so you know what I'm talking about. It's these guys. Oop. Let me do that again. It is these guys right here, right? These are called objective lenses. If you watch and try out the virtual assignments, you can see that it rotates, right? It rotates, it switches magnification. So notice how this one's kind of short and stubby. This one's more of a medium size, and this one's a longer size, right? Because it zooms it in more. So hopefully this makes sense, but I'm going to say it anyway. The shorter one doesn't have much zoom, right? But the longer the barrel is, the more magnification it has. So going back to the other image, this one. So like this guy right here, he's short and stubby, right? So you can still magnify, but not as much as like these two right here, because they're about the same length. So the longer the barrel, the more magnified it is. So here is what that would look like, right? So 10x just means that you magnified it 10 times, right? So this is what it would look like if you magnified it 10 times. If you magnified it 20 times, it looks like that. This is on the same spot, right? So hopefully it makes sense to you that when I zoom in, I lose some of the picture, right? So let's pretend, oh, how do I do this? Let's pretend that it zoomed in on this, right? Because I'm not moving anything. I'm just zooming in as I go. I stay in the same spot, right? So I zoom in, I get this one now, right? And then I zoom in again, then maybe it looks like I'm grabbing this one. So then I get this image, right? And then I zoomed in again, and all I can get is, I don't know, like this little piece right here, right? So then I get this image right here. So hopefully that makes sense. I know this is really difficult in an online environment because we are not looking at things, we're looking at images. So I'm zooming it in every single time, and I'm staying in one spot, right? So I will lose some of the bigger picture. Okay. The unit that we're going to be using for this one is micrometer. That is what it's called. So a lot of the microscopes that we have in our actual lab classes, and they have a tiny ruler in there, right? Because again, we're trying to measure. So we need a ruler of some sort. Not all of them have that, right? Not all of them have that. So when it doesn't, we need to find a way to calculate it. So that's going to be the next slide. So I'm going to put a star here because these couple slides are very, 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 very important, right? So whenever you take a microscope image, you have to put something called a scale bar, right? So that's these red things that I've circled right here. Those are called scale bars because again, in terms of cells, right, I can tell you that, oh my gosh, that cell is so big, right? But like, how big, right? It's the same thing. Oh, that cell is so small. Like, how small, right? So I need to be able to explain it and give an accurate description and an accurate size of it, because if you've ever bought anything from the internet, right, you need to take a look at the specs, right? As a scientist, you're not going to be like, okay, yeah, that's a big bug, but like how big? Because that might depend on, I don't know, like if you're researching it for habitat, right? You need to know how big it is, right? In an accurate way. So that's what the scale bar is for, right? So it gives you a relative size of the specimen. Because this guy right here, this beetle, that thing is huge, microscopically, right? Microscopically, that thing is huge. This one, right, these cells right here, small. right? How do I know that? Because of the scale bar, right? Because of the scale bar. So here is a real life example that hopefully will make a little bit more sense. The Chick-fil-A cat. Hopefully you guys have seen this cat before. If you haven't, it's pretty old. I don't know how old it is, but it's an old image. But this cat was so small that the owner needed to show the world how small it is, right? So what the owner did was they put Chick-fil-A sauces next to the cat to measure the cat, right? Just to show that this cat is really small. Because if I took away the Chick-fil-A sauces, I'd be like, yep, that's a cat. Like, but I won't actually grasp the idea that this cat is that small. So most people know what a Chick-fil-A sauce pack looks like and how big it is. So they use that in order to tell you, like, yes, this is 2 Chick-fil-A sauces small. So if you want to be technical about it, I can go to Chick-fil-A, right? Get a sauce pack, measure that sauce pack, right? And then multiply it by 2 because there's 2 sauce packs, and then I would get the size of this cat, right? And every semester I forget about this, but it has grown now. I think it's like 5 or 6 Chick-fil-A sauces big. But that's the idea of a scale bar. So the Chick-fil-A sauces, this would be your scale bar, right? You're telling the person, hey, this is how big this Chick-fil-A sauce is, and this is how big my cat is. So then you have that that real visual of, yeah, this cat is very small, right? It's the same idea when we take a look at cereal boxes, right? Lucky Charms. And I know it sounds dumb, but it really does happen. Down here in the very, very tiny corner, see how it says enlarged to show detail? Right? They tell you that because they don't want you to buy the cereal and expect the marshmallows to be this big. They're not that big. They're very small, right? So they're telling you in very, very tiny writing that they zoomed in on this photo just to show you what the marshmallow looks like, but it's really not that big. So it's the same thing again, same idea with the microscope, same idea with the scale bar. They're telling you, hey, this is how big the image is using this microscope, but it's really not that big, right? It's not lifelike. It's not a 1-to-1 ratio of picture, right? So hopefully that makes sense. If it doesn't make any sense, let me know. But this is a really difficult concept to grasp, even with in-person students, and not just you guys, right? But in-person students can't grasp this idea that you have to tell your audience what size your cell is, right? Because if you want to think about it in terms of like tumors, right? Like saying that a tumor is really big means that it's serious. But if you can't give the doctor the size of that tumor accurately, they don't know how to deal with that, right? So this one, super duper important. We need to calculate. So I'm going to show you how to do it. It's not too bad, right? You might want to have a ruler with you though, right? You might want to have a ruler when you do this because your assignment will ask you to measure a bunch of these things, right? So let's walk through it step by step. Take it easy. Maybe watch it one way through and then do it together with me and just replay, right? So the first thing it says here is that the scale bar is 100. So that is, let me change colors. That's this one right here. Like it tells me already that the scale bar is 100, right? Where do they get that number from? It depends on the image, right? It depends on the image. It's just telling you that this size line represents 100 micrometers, right? So if and this is the cool thing about being able to use an iPad, if I do this, that green line represents 100 micrometers. That's what that means, right? And then it says the scale bar line is actually 32 mm long. So this is from a ruler. This one from an actual ruler, right? So I took this ruler, I moved it over there and I measured it. It measures 32 mm long, millimeters, right? So now I can take that tiny scale bar and figure out how big the beetle actually is, right? So I take this ruler and I measure this part right here, right? It says the red line is 83 millimeters long, right? So I know that the scale bar is 32 long. The bug, using the actual ruler, using the actual ruler, is 83 long. So we're trying to figure out proportionally what is the actual size of this bug, right? So I'm going to rewrite this on the next page so that the numbers make sense. So here we go. So what would you do, right? So the first step is measure given scale bar with actual ruler, right? And that number, actual ruler, was 32 That's what I got from my ruler, right? Now, most rulers have centimeters. This is why I taught you conversions first. Most rulers have centimeters, so you have to convert it, right? Or you can use the centimeters. That's fine. I'm not really picky. Just make sure your units are the same all the way through, right? So I did that already. And then the second step is to measure the specimen with an actual ruler. So same thing. That's when I took the ruler, and I know this is difficult because I don't actually have you in front of me. I take the ruler and I measure the bug, right? So that's the green line that I drew right there, which is 83 right? And then I just calculate, right? So I calculate. Here's the equation, right? So the equation is the specimen measurement over scale bar measurement times the scale bar value. And then you would get your answer, right? So if I plug these in, then I have the specimen measurement, that's the 83 Scale bar measurement is 32 right? And then I multiply by the scale bar value. The scale bar value is this one, right? The 100 micrometers. 100 micrometers, right? Because essentially you're talking about how many times does that scale bar go into the size of the beetle. That's what you're doing, right? And then you're multiplying it by the value that it actually represents. So let me get a calculator. So I have 83 divided by 32 times 100. I get 259. I'm just gonna round like that, micrometers, because these units, they cancel, so what you have left is the micrometers. So this is the actual size of the beetle. That is the actual size of the beetle, which is very, very, very small, right? Compared to like a penny, right? Or compared to like, I don't know, like a grain of rice. It's even smaller than that, right? This is the actual size of the beetle. So the assignment that you guys need to do is you're going to have to pretend as if you're drawing from a microscope, right? There's microscope images on there. You can pick whatever you want. I don't really care. You can pick whatever you want and then do the same thing. So let me demonstrate for you an example, and then your homework should hopefully make sense, right? So it's gonna make you print out a paper. I would highly suggest to print out the paper and not just do it on your iPad, because you need to make sure that it's to scale. Sometimes, like if I zoom in and out of my iPad, then it's not as accurate. So I'm gonna record another video talking about the actual assignment. I'm just gonna show you the microscope part, right? So let's pretend this is the image that I chose. So just imagine that you're looking through a microscope and this is the image that you see. So what you're drawing is you're drawing inside this line, right? This one. So don't include this blue one right here. So your main job is to, as accurately as you can, represent that same image on your drawing. And again, we're not grading you on artistic ability, so you don't have to worry about, it doesn't look that great. It's fine. It's not a big deal, right? It's not a big deal. So what I would do with my printed out paper is I would try to draw that to the best of my ability to scale, right? So what does it mean to be not to scale? Just in case you're like, I don't know what that means to scale. To scale, or sorry, other way. Not to scale means I'm drawing it like this. That is not to scale, right? Because those are two different things. I need to draw it exactly as I see it from the picture. So the best that I can, right? Like, sorry, this is going to be really difficult because I can't, or actually, you know what? Here. Let me do this. Let me put it in here so that you can see it. So this is the image I'm drawing, right? So let me scooch this over. So this is the image I'm drawing. So I'm going to try to draw it as accurately as possible. So I can't even draw it like this because that's too big. That is not to scale because it's taking up way too much of that space, right? So as best as you can, try to get it to match, right? So then, I don't know, like about like this. Right? And again, don't worry about like artistic ability or whatever. Just as best as you can. Size is really what I am looking for here. So something like that. And then this one kind of looks like that. Even that, I probably would have gotten it bigger because see how much bigger this space is than this space, right? So that's really what you're practicing. You're practicing drawing it so that it's as accurate as you see it on the microscope, right? That is the main idea. So again, I would probably draw this bigger. So if I was measuring this is just pretend, right? So if I was measuring this was an image. It's not on here, but the slide I got this from, the scale bar was 15 micrometers, right? So this is just me. I'm just pretending. So don't copy mine because I don't have an actual ruler next to me. So what I would do first is I would measure the scale bar. So I measure this one. So I'm going to pretend it says 1 centimeter. I'm just pretending here. 1 centimeter. Or sorry, that goes on the denominator. Right, because scale bar goes in the bottom. So I measured the scale bar with an actual ruler. I got 1 centimeter. If I measured this cell right here, let's say it's about the same size as the scale bar. So maybe it was 1.5 centimeters. That's this guy, 1.5 centimeters. This one was 1 centimeters. And then the scale bar number that it gave me was 15, right? So then these two would cancel. Let me get my calculator. 1.5 divided by 1 times 15. I got 22.5 micrometers. So the actual size of this cell right here is 22.5 micrometers. Right? So that's going to be a part of your homework. So if you need to watch it again, if you need more help, let me know and just send me an email. I will try my best to show you how to do it. But hopefully that makes sense, right? So you're just telling me what does this cell actually look like and how big is it, right? How big is it? So if you have any questions, just send me an email. Otherwise, hopefully that makes sense. And I'll see you guys probably in class.